Mike, I'm really glad you brought up both starting materials and cell banks. We're going to get to both of those in more detail, specifically with starting material. When it comes to quality and consistency of raw materials, acquisition of those starting materials, it's obviously critical to the success of the therapy. Most cell therapies today are produced from a single patient at a time, using that patient's cells as the starting material. That one dose at a time approach can be difficult. And so what I'm asking both of you, what are the specific roadblocks for autologous when it comes to starting materials? Mike, I can start with you if you'd like.
I tell people in the industry, if we just look at it very basically, I'm making a salt solution, let's say sodium chloride and water. And the benefit of that is we know exactly what that sodium chloride is, right? NaCl, 99.9999% pure. What if it wasn't? What would we do? We'd have to make a lot of batches of salt solution to characterize it. It's the same thing with autologous therapies. Every patient is different. Tumor microenvironment different. If for TIL, the apheresis could be different for traditional like CAR T. And that presents a burden of characterization, which is why I'm a real proponent. We have to really think about this from the very, very beginning as we develop these processes, understanding that there will be post-approval changes. So how do you manage that from a regulatory perspective, right? It means we need to test a lot of patients. That's in a nutshell, the way I look at
it. Good. Okay. John, I want to get your perspective too on starting materials and maybe some of the specific roadblocks from an autologous perspective.
Yeah, I mean, I think dovetailing on what Mike has said, what effectively what he's describing is variability or heterogeneity of that starting product, right? The cells, across different patients as they're collected in apheresis, patient A may have seen in some cases 6 lines of prior for some of these clinical trials, and the T cell compartment there is effectively at the least compromised, and at the worst potentially destroyed. And so, trying to make an efficacious cell product from that shoddy starting material is in many cases difficult to do. And of course, we probably should have led with. From a supply chain perspective, both plasmid DNA and vector are both at present limiting with regards to gene modified cell therapies. These are both at present, in most cases, backlogged, and companies are waiting on them. There have been a few articles of late. Wall Street Journal, I know, published an article not long ago, basically that there are patients who are dying while waiting on their therapy simply because there's not enough manufacturing capacity worldwide to treat these patients. So the starting material then becomes extremely important if that's the rate limiter, and vector and pDNA are both part of that. But returning back to where we started, it really does come back to this heterogeneity and understanding, how to normalize that. And I think there's a couple potential ways to mitigate the risks that come with some of this variability. One is, and we're seeing this already, as these therapies are approved, they're moving into earlier lines. And so, the starting product then gets hit less with things like radiation and chemotherapy prior to creating cell therapies. There's the concept of at-risk banking. So when a patient comes in on day one and he or she is diagnosed with some sort of disease, at that point, that person could get an apheresis and then have that stored for the appropriate time to create a cell therapy. Now, that obviously creates a whole logistical, cold storage infrastructure question, which we won't get into here, but it's certainly an option. And then, lastly, and I think this is worth pointing out, there are companies out there at the moment that are retrospectively looking at data from patients who have done well on these therapies, and they're defining the characteristics that are prognostic or otherwise predictive of response. And that'll enable manufacturers then to start building those features into those products over the long term.
Okay.
Those are, you hit upon two very good points. And the first one is that starting material in the clinic could be very different than what you see commercially. We're starting to see that as these approved cell therapy products are surveilled in the industry. The other one is this, as you said, I call it the holy grail of cell therapy, is to predict the outcome, right? This is where the role of translational comes in, and looking at biomarkers and things, and being able to really understand what characterizing that starting material means. It's also indicative of where we are technologically or scientifically in the industry, where we have a lot more growing to do. One note on scheduling for the autologous products, and given the fact that patient, we call vein to vein or arm to arm time, or whatever you want to call it, is very important. That relates to the scale out strategies, right? Where typically in a monoclonal facility, when I used to work in monoclonals, we were talking about 95% plus equipment utilization, meaning that equipment is making product 95% of the time, and 5% of the time is down on maintenance or other issues. For cell therapy, for autologous, because we have to respond to the needs of the patient and the scheduling, if we get above 50% equipment utilization, we're golden in a commercial environment. So so you have to really understand what that means for your organization. Does that mean, and this is where automation comes in, you have 25 operators sitting waiting for the patient, only 12 of them in use at any one time, or do we use automation to help with that equipment utilization issue? So those are some very good points. I would love to be, I wonder what it'll be 10 years from now with regard to starting material and how it can predict an outcome of a cell, or at least a cell culture process. It'd be interesting to see what'll happen.
Yeah, probably light years different from where we are now, I would think. I want to make sure we're having the same parallel conversation around donor cells, and from a scale perspective, let's talk a little bit about the allogeneic side of this, and from a potential, where does the potential lie, and what are the challenges? Let's talk from a donor perspective. Same type of conversation. John, you can.
I'm sorry, I missed the question.
Oh, sure. Sorry. So from a donor cell perspective. Oh, okay. Sorry. Can you hear me now?
Yeah.
Okay, good. John, I'll start with you. From a donor side, I want to make sure we're having a parallel conversation and talk about it from an allogeneic perspective. What are the biggest challenges when it comes to donor cells for allogeneic therapies? Where does the potential lie?
Yeah. It's a quite a contrasted conversation around allo when we're talking about donors, because typically there's some level of gene editing that needs to occur. And as a result of that gene editing, you've got, at least in my eyes, two primary concerns. One is around just safety, and we can talk about that in a moment. And then the other is the resultant scale that you can achieve as a result of whatever gene editing that has been done. Typically, without getting too much into the weeds with regards to the science, but the intent of the gene editing is to eliminate allogenicity, whether that be graft versus host disease or just general immunogenicity and rejection of the drug product. But from a safety perspective, after that gene editing is performed, we as manufacturers need to ensure that the genetic stability and otherwise the genetics integrity of the resultant cellular population is indeed safe, and we're not otherwise subjecting the patient to additional risk through genomic abnormalities. From a scale perspective, and I think I heard somebody else on your other podcast, the CEO of Poseida a few weeks ago, Aaron, he was talking about I forgot what he termed it as, but it's it's something that's very relevant, especially in the world of T cell therapies. We're knocking out the TCR to eliminate graft versus host disease, and how 99% of T cell therapies are expanded to scale is through engagement of that same TCR. So when you knock that out, there need to be other ways of expanding those products. And companies like Poseida have already developed these strategies, but it is one that may be suboptimal, at least compared to what we're currently doing with CD3 beads. And then I guess the other component is understanding, and I think I mentioned this at the top of the webinar, as we start to expand these cellular populations into the 10 to 11th, 10 to the 12th, there is the risk of functional exhaustion, and navigating how to get that expansion without compromising the functional integrity of that cellular population is an area that a lot of companies are looking into at the moment.
Gotcha. Okay. Before we move on to aseptic processing and that topic, I want to make sure, Mike, I'm giving you the opportunity to chime in as well on, from your perspective, the biggest challenges with regard to donor cells.
I mean, I think that John hit the major points. Now, my expertise is in manufacturing and not in designing these types of products. However, the TCR knockout issue is I think was something that was in my mind. Also, the genetic stability of your construct as you manipulate these cells, I think, is something that has to be investigated. How we perform those genetic manipulations, but also when we look at the donor, the typing of the genetic type of the donor cell itself, I think has a lot to say about success rates. But this